เคสมัยทุกคนยังไม่มาผมพูดไปเรื่อยๆกันสมัยนู้นผมเป็นนักเรียนเตรียมแพทย์เงี้ยตึกอาจารย์ตังที่วิทยาศาสตร์การแพทย์ That's me okay this my class สมัยนู้นเตรียมแพทย์เชียงใหม่เพื่อเข้าเตรียมแพทย์ได้ก็ทิเก็ตเรียนแพทย์เชียงใหม่แต่สมัยนู้นโรงเรียนแพทย์เชียงใหม่ยังไม่เพิ่งเปิดแต่เตรียมแพทย์เชียงใหม่เรียนอยู่ตึกอาจารย์ตังที่ถนนศรีอยุธยา We always have a party with our teacher the chairman department of physiology invite us for dinner function at night so we always go there for free dinner จำได้เปล่ารูปอย่างนี้นั่งรถไฟมาถึงเชียงใหม่ Welcome freshman That's me นักเรียนแพทย์สมัยนู้นใส่ชุดสีขาวแล้วก็ใส่หมวกสีเขียวเนี่ยลงจากรถไฟ Get off the train arriving to เชียงใหม่ campus from Bangkok หน้าหอชาย the boy dormitorium สาวๆรุ่นผมนะฮะ หน้าหอหญิง Party again Welcome freshman รุ่นหลังโอเคเมื่อวันจันทร์ใช่ไหมแผนกคณะแพทย์ก็มีลำวงสมัยก่อนนั้นพวกผมเต้นบัลเล่ครับเพื่อหาเงินเข้าคณะแพทย์ ผมเป็นคนถ่ายรูปแล้วอาจารย์วราวุธเคยได้ยินชื่ออาจารย์วราวุธเปล่าเขียนเพลงไงฮะวราไม่ใช่แกเป็นคนสอนบัลเล่ graduation และอาจารย์รู้จักจำอาจารย์พวกนี้ได้เปล่ารีไทร์หมดแล้ว สมัยรุ่นผม 1967 the girl class 9 คนฮะรุ่นผมโอเค This picture is Dr. Stang who pioneered in build up basic medical science in Thailand เอาเงิน Rockefeller Foundation to start basic medical science ถนนศรีอยุธยาส่งคนไปเรียนเมืองนอก use the funding from Rockefeller Foundation so a lot of basic science teachers today build from his thinking like chemistry biochemistry อาจารย์หมอระเบียบผู้อำนวยการโรงเรียนแพทย์คณะแพทย์โรงเรียนแพทย์เชียงใหม่สมัยนู้นอาจารย์บัวเรศทองคำ the first president or อธิการบดีมหาวิทยาลัยเชียงใหม่รุ่นแรกหมอทองท้วม our graduation right here that's me how skinny is it this is our crowd went to US ขึ้นเครื่องบินไปมาเนี่ยโตเกียวทาวเวอร์ผมอยู่นี่ฮะเดี๋ยวจะให้ดูนี่คุณหมอมณิษฐ์แสนมณีชัยคุณหมออรินีอยู่ที่ไหนนั่นลูกสาวแกจะมาช่วยทำ clinical skill วันพรุ่งนี้reunion คืนสู่เหย้า1992 2535อาจารย์หมอมนตรีทองต้วมนรีศรีบริบูรณ์ He's the head of the Illinois project We have affiliate Chiang Mai Medical School and Illinois and this คุณหมอบุญสมมาติน the first dean of Chiang Mai Medical School That me That me we went to รักษาพวกแมวโอเครู้จักหมอเกษมวัฒนชัยเปล่าโอเค The one in my class the fourth graduate he's highly success องคมนตรีสมัยนั้นหัวยังไม่ล้านใช่ไหม The most high rank officer from my class you bet
Hi hello and welcome to the fourth day. So, just like to remind you a couple of things. First, for those of you who sign up for the certificate of attendance, I'd just like to remind that you have to be here 4 out of 5 days. Okay, so please make sure to be here today and tomorrow also. And also tonight, right after our last sessions for today, we have a party, so everybody's welcome. There will be karaoke, I heard. Of course, food. I don't know if there's drinks, but yeah, you'll be here and get a chance to talk with our speakers on a more of a personal note. So there will be an in-depth and very informative thank you, discussions. Ngong sang fai ah, boleh. All right. I would like to introduce our moderator for this morning's sessions. Dr. Paul Doughted. Okay. He received his PhD in marriage and family therapy at the Nova Southeastern University, that in Fort Lauderdale, Florida. And right now he's an assistant professor of clinical pediatrics at the Division of Developmental Pediatrics, Department of Pediatrics, and at the Texas Tech University Health Sciences Center, that's in Texas. So, Professor Doughted.
Good morning. It's with great pleasure that I'm here and able to introduce we'll be introducing to you the next two speakers this morning. First of all, just on a personal note, I would like to thank all of you for attending and being a part of this conference. I have enjoyed being able to spend time in Thailand since 1992 on 4 different occasions. And not only the country is wonderful, the food is very good, and the people are very warm and appreciative. And I want to thank you, as I said, for being here today. And I also like to bring greetings from my chair, Dr. Richard Lampy, in the Department of Pediatrics. He is, prior to coming to our department as chair, he was a career military officer and pediatrician. And he and his wife, many years ago, in fact, adopted a young child from Thailand. And so he has some very, very warm memories of Thailand. But from him, welcome. I was telling a friend that sometimes it's easier to introduce people that you've just met or that you don't know very well than people that you've known for a long time. And so this morning I will have the opportunity to try to introduce 2 people that I've known for a long time who have been at the medical school at Texas Tech for many years and have done many wonderful things over those years. To start off with, though, I'd like to introduce my good friend Elmus Beals. Elmus is currently an associate professor in the Department of Cell Biology and Biochemistry. He actually joined the faculty at Texas Tech University School of Medicine in the fall of 18. He's not that old yet. He joined the faculty in 1984, so he has been there many years. He currently lives in Lubbock with his wonderful wife, and he has 2 adult daughters, one who lives in Japan currently and who he will go spend some time with on the way home. Elmus received his doctorate in 1977 from the Department of Cell Biology, Baylor College of Medicine in Houston, Texas. As I said, one of the difficult things in knowing someone for so long and introducing is that sometimes you forget the details. So I've given you some details, but one of the things that is to me, most impressive about his curriculum vitae is that over the years he has helped and worked with many young students to get their masters and/or their doctorates. He's worked. In fact, he worked many years ago with one of my colleagues, and during the summer, and helping him do some research. And so he has a real heart for working with students. And besides that, he's a great guy. And this is his fourth time to be in Thailand as well, and I think second time to be in Chiang Mai. Dr. Elmus Beals.
Is that me he was talking about? I don't know, I may just end up sitting down here, but I will try it standing up for a little bit. Good morning, Sawadee krap. And thank you, Paul, for a very nice introduction. So, Dr. Siriwan has given me the charge of reviewing molecular biology and biochemistry in 2 hours. Huh. So I would like to reiterate something that Dr. Tinner said 3 days ago, and that is the key for you is to probably buy one book and practice, practice, practice. So we can just kind of scratch the surface this morning. I'm going to try to hit some highlights, what I think are some of the fundamental things that you need to know, especially about molecular biology. I want to make sure that you understand how DNA is organized, how genes are organized on DNA and in chromosomes, how genes are expressed and converted into some RNA or protein, and how that's regulated. I think those are really the fundamentals. But I have to warn you that just for lack of time, we're going to have to omit many important things. So that said, let's begin. Okay. This I'll just sit down and do it this way. So I've divided the presentation into a number of topics, and we're going to begin right now with just the overview of the genome and how it's organized and how it's expressed. So let's try a number of questions here in the beginning. I'll start with this one. After treatment of chromatin with micrococcal nuclease, the majority of DNA is protected from digestion. It remains as double-stranded fragments bound to which of the following? A, histone H1, B, nuclear membrane, C, nucleolus, or D, an octamer of histones. So do you have your polling devices? If you do, please vote now. No? So only one person has a polling device? Is that the deal? It's okay? Okay. That's going to complicate some of these things. The feedback is great to know where you stand. So the one person that voted got it right. How about that? So let's just assume that everybody who didn't vote got it wrong. So let's go through a brief explanation. What we're talking about here are nucleosomes, and they were originally observed with an electron microscope. As in this bottom picture right here, it looks like beads on a string. The string is a piece of DNA. Maybe I should pause a minute and let those get handed out. it seems that the polling devices are suddenly more important than the DNA. Okay, so again, the bottom picture here, beads on a string is the way these structures were seen with an electron microscope. This is probably 30 or 40 years ago. Scientists originally did not know whether this was an artifact or not, but it turns out it's not an artifact, it's something that really exists. This fiber right here we call the 10 or 11 nanometer fiber, and it can be coiled up into a larger fiber that looks like this. And the next slide illustrates a cartoon to explain what we're looking at. This 11 nanometer fiber, beads on a string, form of chromatin right here, shows the way histones create a little barrel-like structure around which DNA is wrapped twice, and a linker piece of DNA to the next nucleosome. So in the original question with micrococcal nuclease tends to digest the DNA that's accessible between these nucleosomes. That's the structure that was seen in the electron microscope, but this structure, like winding up a rubber band, can be made into a coiled coil kind of a structure, this 30 nanometer structure right here. So that relates, I'm going to go back to the previous slide. There are the 11 and 30 nanometer structures. So micrococcal nuclease will digest the DNA between these beads on the string. So this 30 nanometer structure for mitosis can be packaged into denser and denser loops, and the loops are looped, and that can be then, that's a visualization of this mitotic chromosome where DNA is highly compacted, but in interphase it's more like the 11 and 30 nanometer fiber. So there are genes along that in various places. So the challenge for enzymes then is to copy those genes into proteins, and somehow they have to read through those nucleosomes. How do they do that? Well, I don't really know, but it happens somehow. So here is a different view of the nucleosome, and it seems very likely that you will see questions about this organization on the USMLE. We can't be certain, of course, but what you need to know is that the nucleosome is made up of 4 histones, each present in 2 copies. Their names are histone H2A, H2B, H3, and H4. So 4 histones, 2 copies apiece, make this, it's an octamer. And the DNA is wound twice around that octamer. So here's a side view here and a front view right there. A 5th histone, called histone H1, this guy right here, is associated with a linker DNA in between the nucleosomes. And so that's all packaged together to create the beads on a string. Okay, next question. Which of the following is true during RNA synthesis by DNA-dependent RNA polymerase? A, inorganic phosphate is cleaved from the precursor nucleotides. B, only one of the strands in a chromosome can serve as a template. C, nucleotides are added to the 5' end of the growing chain. D, there appear to be specific sites on DNA for initiating and terminating RNA synthesis. And E, the polymerase exhibits an active proofreading function. So if you're ready, go ahead and vote on this. Good. So the answer is D. So 50% of you got it right, but that means 50% got it wrong. So it's probably worth going through an explanation for the half that got it wrong. Let's try this one. Failure of an endonuclease to recognize the sequence AAUAAA in the 3' end of heterogeneous nuclear RNA will cause a defect in which of the following processes involving RNA? So if you're ready, go ahead and vote on that one. Okay, that's all over the map. Well, that shows that it's worth going through an explanation of these things. So those two questions prefaced the big picture of the concept of gene expression and how it's regulated. Now, what is a gene? A gene is a region of DNA that is transcribed into an RNA. So that RNA could be transfer RNA or ribosomal RNA or messenger RNA or a microRNA. But the main thing that we're talking about right here is how is messenger RNA made. Messenger RNA, the RNA, of course, that encodes proteins. That's probably the more complicated kind of gene expression, so let's focus on that. So first, just the organization of genome chromosomes. Now, we've already talked about the concept that the DNA is packaged in nucleosomes. So layer that on top of what we're looking at here. Here is a hypothetical example, human chromosome number 22. It has 48 million nucleotide pairs from one end to the other end. And if we zoom in on a small fraction of that, just 10% of the total, it's a region that contains about 40 genes. And if we look even closer, zoom in on that tenfold, there's a region that contains 4 genes. So this red and brown represent separate genes. And finally, let's just zoom in on this gene right here, a single gene. It's organized, as you probably are aware, into regions that end up in messenger RNA and regions that are spliced out, called exons, the red areas here, and the introns are the intervening sequences in between. So in this example, this first exon, this red region, represents the first part of the transcribed portion of the gene. The first nucleotide of that exon is the first nucleotide that ends up in the RNA. And at the other end, the last nucleotide ends up as the last nucleotide in the RNA. And upstream, in this yellow region up here, are regions of DNA that control whether the gene is converted into RNA or not. So how does that work? Now, one of the things that in 2 questions back it asks you to recognize is whether or not genes are on only one strand of the DNA or both strands. And this illustrates the concept that both strands of DNA can serve as template. Here, for example, the top strand, we'll call it, goes from 5' to 3' from left to right, whereas the bottom strand goes the opposite direction, from right to left, 5' to 3'. And these regions on the top strand show 1, 2, 3, 6 or 8 genes, and on the bottom strand, a number of genes as well. So somehow, punctuation is used. It's like reading a book or something like that. Helps RNA polymerase know which strand to copy, which direction to go, which strand to use as template. So we're going to kind of focus on how that works. So the big picture is this. Here's, again, an example gene in a region of DNA with introns and exons. From end to end, that DNA is copied with RNA polymerase from the first nucleotide to the last nucleotide into an RNA. Now, the beginning of the process, initiation of RNA synthesis, is very precise. It begins at a precise nucleotide at the 5' end of the gene. But at the other end, when RNA polymerase reaches that point, it actually keeps going. RNA polymerase doesn't recognize when to stop, it keeps going. But there are some post-transcriptional processes that then add a cap to the 5' end, chop off the 3' end, and add a tail to it. That process also removes these introns, these yellow regions here, to generate a mature RNA that has a cap on it and a run of perhaps 150 to 300 A residues. That's all added after transcription. So the question becomes, how does RNA polymerase recognize where to start, and how does the polyadenylation machinery recognize where to end? Here's an example over here on the right of some of the amazing variation that one can obtain in the kinds of proteins that are made just by varying where transcription starts and which intervening sequences are removed and where the 3' end of the RNA is. So for example, this shows the alpha-tropomycin gene. In striated muscle, there is this kind of a transcript that's made. It's different in smooth muscle, fibroblasts, and brain. And the differences have to do with what is used for an intervening sequence or an intron and what is used for the 3' end. So somehow this is all regulated in a remarkable fashion. People are only just now beginning to understand how it works, but the fact is that it does vary, and so it would generate a variety of different proteins. Now I want to focus just a little bit now on the initiation of RNA synthesis, because that is probably the key regulated event, not the only, but the key regulated event. So here is an example of a diagram of a gene. The start of transcription would be right there at the beginning of the red. Upstream from that, there is DNA, an important part of which is called a Tata box, typically about 30 base pairs upstream. And there are other regions, such as this so-called enhancer, where other proteins bind. So proteins bind to the Tata box and enhancers and silencers and other important DNA upstream of the gene, and will create an enormously large complex of proteins that center around RNA polymerase. So this light blue region right here is RNA polymerase. Now, RNA polymerase by itself can't recognize where to start. It's all of these other proteins that bind, for example, the Tata box and the activator region upstream, and assemble a huge complex of other proteins. Together, they are able to precisely tell RNA polymerase which nucleotide to start at and which strand to copy. And then once transcription begins, the RNA polymerase unwinds the DNA, proceeds in a 5' to 3' direction, and elongates the RNA. So that brings up an important concept. RNA synthesis is collinear with DNA synthesis. Always goes 5' to 3'. Not only that, when the code in messenger RNA is translated into a protein, that's also read from 5' to 3'. So the protein is synthesized from its N-terminus to its carboxy terminus, reading in that 5' to 3' direction. And so that's an important concept. So that is a big picture of how transcription is regulated at the level of initiation. This is the big picture of how transcription is regulated at the termination site, at the very end of the messenger RNA. The most common punctuation or word that the polyadenylation machinery recognizes is AAU, AAA. There are exceptions to that, but that's still the most common. That is typically about 10 to 30 nucleotides upstream of where a cut is placed in the RNA and where, so the cleavage occurs right there, and then the addition of the 250 or so adenine residues occurs. Is that clear? Because I'm going to ask you some more questions now and see how you do on that. So that was hopefully a description of the background behind those two questions that I asked earlier. Okay, so now once the RNA, a messenger RNA is made, it has to be copied into a protein. 5' to 3' is the direction. And to understand that, we need to understand the concept of codons and how the RNA structure is arranged. This is a typical messenger RNA, at least a diagram of it. At the left-hand side here is the 5' end, or the head end of the RNA where the cap is placed. This region right here, or this point right here, is the start codon, the first methionine codon, the AUG. So a ribosome would bind to this 5' UTR, stands for untranslated region. It will bind somewhere up here at the 5' end and slide along the RNA looking for the first AUG. And there it will begin translating a protein, and it keeps track of that reading frame until it reaches the first stop codon. And that, from the start to the stop, is called the translated region. But all messenger RNAs, in humans at least, in eukaryotes, have a 5' untranslated and a 3' untranslated region. Okay? So let's see how you do with recognizing and understanding the concept of codons here. So here's a question. The following numbered codons relate to the partial sequence of an mRNA for a protein. Termination codons are AUG, UAG, UGA, and UAA. The initiation codon is AUG. The triplets are numbered as shown. So this shows 132 to 139 right here. Insertion of uridine between the end of codon 135 and the beginning of 136 will result in a protein containing how many amino acids? So the insertion that we're adding is right there. Right there. Let me give you a moment to think about that. Okay, go ahead and vote. Well, it looks like I gave most of you enough time, but only got 18 votes, so sorry. That took some calculation and thinking about that, but good job. So the answer is E, 138, and the logic is by adding, if we look up here, adding a U right there, you just recalculate the reading frame. And if you understand this concept, that means you understand the concept of reading frame. So it would read UAU, then immediately UUU, AUG, UGU, UAA, and UAA is now a stop codon. That stop codon right there. So that had slid over into position 139. So if 139 is a stop, then that means there are 138 amino acids. So good job. New topic. The structure and replication of DNA. Now we've already touched on this just briefly. Let's see how well you understand it here. So this question is, what is the complementary sequence for a DNA strand having a sequence of G, C, T, A, G, C, C, C? If you're ready, go ahead and vote. Yeah, it's a voting. The correct answer is C. So let's see, why did you get that wrong? Let's think about that a little bit. So what we're doing here is testing whether or not you understand what 5' to 3' means and whether or not you understand that the top and the bottom strands are running opposite directions to one another, okay? So this strand that we're given right here with that little P right there indicates the 5' end. That's a phosphate group. And the phosphate group, at the 5' end of every nucleotide, there's always a phosphate group. So that's how we know that we're reading from 5' to 3'. Well, if that is 5' to 3', then its complementary strand, 5' to 3', ought to be right down here below it, and it ought to read G, 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 C, T, A, G, C, which is answer C. Do you follow? Anybody want to talk about that a little more, or are we okay? Okay, we're okay, good. Okay, with that concept in mind, that DNA, the double-stranded DNA, we often represent it as not a twisted strand, but as a top strand and a bottom strand. A couple of important points that you need to know, and I bet you already know. The first is that replication is semi-conservative. What does that mean? Well, that means that here is the original DNA strand, and when it is duplicated, one strand goes to the new daughter strand, the other goes to the second daughter strand. And that process continues. The daughter strands receive one strand or the other of the original DNA. The second major point to understand is that DNA synthesis in cells that are actively growing, on each chromosome there are many, many what we call origins of DNA replication. And so over here on the right at the top, I'm trying to represent one origin of replication. It's a unique sequence of DNA that the DNA synthesis machinery recognizes and binds to and begins the process of DNA synthesis. The two strands of DNA are separated, and although it's not showing it here, what's going to happen is that DNA synthesis goes in both directions. It goes to the right and it goes to the left. There's a primer that's added. The primer is elongated. The same thing happens on the other strand, and now this so-called replication fork, there are two of them that proceed in opposite directions from the replication origin. So the important thing to understand there is the complication between the direction of the synthesis. Which way is DNA synthesized? Always 5' to 3'. So that means that it's using the 3' to 5' direction of the other strand as its template. So in this example right here, the top strand, let's say, is reading left to right, 5' to 3', and the bottom strand is reading right to left, 5' to 3'. Well, that creates what we call a leading strand here going to the right and a leading strand here going to the left that are being synthesized 5' to 3', because they're reading the 3' to 5' part of their template as the DNA opens up. But there's a lagging fork on each strand as well, this one and this one. It shows down here in a little better detail. The leading strand is just synthesized without any breaks, but the lagging strand is synthesized as many little short fragments that are then spliced together. Those are the so-called Okazaki fragments, and Dr. Okazaki from Japan discovered those and won a Nobel Prize for figuring out how that process works. Okay, I think I can quit belaboring that point. I'll bet you already know it. New topic. genetics. So Dr. Graham yesterday, in her talk about pathology, presented a number of genetic diseases, and she emphasized the importance of you reviewing some genetics. So let's review a little bit right now. And we're going to do it in the context of several questions. So let's begin with this first. A young male is being evaluated for developmental delay, mild autism, and mental retardation. Physical examination reveals the boy to have large everted ears and a long face with a large mandible. He is also found to have macroorchidism, or large testes. Extensive workup reveals multiple tandem repeats of the nucleotide CGG in his DNA. Which of the following is the most likely diagnosis for this patient? Fragile X syndrome, Huntington's chorea, myotonic dystrophy, spinal bulbar muscular dystrophy, or ataxia telangiectasia. If you're ready, go ahead and vote. Good. Now I put this question in here specifically to allow me to talk about the concept of triplet repeats. And again, Dr. Graham mentioned this yesterday. So yes, it's Fragile X syndrome. That seems to be a commonly asked question. That's a very common, I guess it's the most common form of genetically determined mental retardation. But one of the unique things about this entire question is that the top 4 choices are all versions of diseases that are caused by expansions of 3 nucleotide repeat units in certain genes. The only one that's not is choice E, ataxia telangiectasia. It's the only one of the 5 that's not caused by a triplet repeat. So I promise you, you're going to see this on the USMLE. So you need to have in your head a general understanding of what this is all about and which genetic diseases are caused by these triplet repeats. Now the causes vary. Sometimes these triplet repeats are within the coding sequence of a gene. So you end up with polyglutamine or something like that within the protein, and that affects the function of the protein. But sometimes the triplet repeats are in regulatory regions of a gene, not a part of the protein. Sometimes they show up in intervening sequences that are spliced out. And so in many cases, scientists don't understand why the disease occurs, just that it really does. Why triplets? I don't know. You know, why not dinucleotide repeats or single nucleotide repeats? That's another question that's not really resolved. But the fact is that there are several of these with trinucleotide repeats. So let's talk briefly about the details for Fragile X syndrome. The gene is called FMR1. Normal individuals at a certain place in the gene have somewhere around 40 repeats. We all do. But perhaps it's because when DNA polymerase is copying the gene, it gets confused and loses its place. Sometimes as gametes are made and the DNA is replicated, the number of repeats can be expanded, let's say, to between 50 and over 200. Well, that individual may not be affected, but it increases the chance that the offspring from that individual might be affected. Those who have the disease typically have, in this particular case, greater than 230 copies of the gene. So, few repeats, normal, many repeats, increased risk. many more repeats disease. That brings up a common theme among these kinds of diseases, and that is of anticipation and increase in severity with generations. If a father is affected with one of these diseases, let's say Huntington's disease, but mildly affected, his offspring have an increased risk of being more severely affected at an earlier stage in their life because they have the risk of having more repeats in their Huntington's chorea gene, if that makes sense. So, just to summarize briefly, triplet repeats cause many genetic diseases. The more triplets, the worse the disease, the earlier the onset. So anticipation and severity. Okay, next question. A 24-year-old male has an autosomal dominantly inherited disease. The patient and his paternal grandfather show evidence of the disease, but the patient's father is asymptomatic. This is an example of A, polymorphism, B, mutation, C, variable expressivity, or D, reduced penetrance. If you're ready, go ahead and vote. Okay. Let's talk about that briefly. I guess I could do it like Dr. Strauss did yesterday. Oh, that's disappointing. So let's think about the clues that you're being given. So the affected individual is the son. His grandfather on his father's side also has the disease, but his father doesn't have the disease. So, and we've already been told it's autosomal dominant. So that means what? An autosome, most chromosomes are autosomes. You have either an autosome or a sex chromosome. So if an individual has this gene, they should have the disease. His grandfather has it. He has it. His father has to have the disease, has to have the gene, but his father doesn't have the symptoms. And it's autosomal dominant. So the only way that you can have that condition is by a phenomenon that in general is difficult to explain, but it's called reduced penetrance. Penetrance is the ability for it to, I guess you would just say, penetrate and become evident. So somehow in his father, it didn't penetrate his skin and show up as a disease. It did in his grandfather and in him. Okay? Next question. A 4-year-old boy presents to the physician's office with coarse facies, short stature, stiffening of the joints, and mental retardation. Both parents, a 10-year-old sister and an 8-year-old brother, all appear unaffected. The patient's mother is pregnant. She also had a brother who died at 15 years of age with similar findings that seem to worsen with age. She also has a nephew, her sister's son, who exhibits similar features. You suspect a diagnosis of Hunter syndrome. The most likely pattern of inheritance for this condition is autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, or none of the above. I'm going to give you a little time to think. Oh, I guess the clock has started. Well, good. So almost half of you got that right. So this is kind of, it's a little bit complicated, so let's just talk through it and see what we can learn about it. So we already know now that the answer is X-linked recessive. X-linked recessive. So let's see, the 4-year-old boy, so by the way, Hunter syndrome is a lysosomal storage disease. I guess it really doesn't matter in this particular case, but it is caused by mutation in a single gene. And of course, it's an X-linked gene. So the patient is a 4-year-old boy. So both of his parents, his mom and his dad, a sister and a brother, are unaffected. His mother is pregnant, but she has a brother who died at 15 years of age with similar findings. So we know that it comes from the mother. The mother had a nephew, her sister's son, who exhibits similar features. So it seems to always come from the mother. It doesn't seem to affect the females, which would be consistent with the idea of X-linked recessive, because the females would have 2 copies of an X chromosome and most likely have 1 normal copy. So we're seeing only the boys are affected and only the sons of women. So women seem to be the carriers. Make sense? Okay, a little more genetics practice here. A 23-year-old female presents with progressive bilateral loss of central vision. A detailed family history produces the following pedigree with filled symbols indicating affected individuals. Which of the following transmission patterns is most consistent with this history? A, autosomal recessive. B, autosomal dominant. C, X-linked recessive. D, X-linked dominant. Or E, mitochondrial. Okay, if you're ready, go ahead and vote. Oh, I guess that's, well, good. So the answer is E. 37% of you got E, which means that 73% of you got it wrong. So let's work through this. So here's an affected. It's a woman. So I'm first thinking, gosh, is this just women? Oh, but there's a man, a woman, a man, a woman. Now wait a minute, look. All of the offspring of this woman right here have this condition. A male, a female, a male, a female. Well, let's see, do the offspring of any of those children have it? Well, this male, none of the offspring have it. This male, none of the offspring have it. This female, all of the offspring have it. So what is the only thing that would be transmitted just through women, regardless of whether or not it's a male and a female? Now, we just saw an example of X-linked recessive, and X-linked recessive, we probably wouldn't see that in very many females, and yet here we see it in females and males with almost equal frequency. So we all inherit our mitochondria from our mothers. So the mitochondrial DNA is what is going on here. This is mitochondrial inheritance. The disease that we're talking about right here is actually, you don't need to know what the disease is to guess correctly what kind of inheritance we're looking at, but this is Leber's optic atrophy, and it's caused by a deletion or mutation in the respiratory chain, and some of those genes are encoded by the mitochondria. And exactly why that occurs, I don't think anybody knows, but this is a bona fide form of human genetic disease. Okay, I think that ends, at least for now, the topic of genetics. In the second hour, I'm going to present a number of, we're just going to go through example questions in the second hour. And so there will be some more opportunity to look at some genetics at that point. What I think, do you want to take a break now, or?
Would that work well?
I think so. Here is the next topic, tools and methods in molecular biology. Why don't we take a break right now, 5 minutes, and then come back and resume.
All right, so we'll see everyone back in about 5 minutes.
New topic now, tools and methods in molecular biology. Most certainly that you will see some questions that require that you know something about this. So, first question. Which of the following statements concerning the introduction of recombinant DNA into bacteria is false? When a bacteriophage is used to introduce DNA into bacteria, the technique is called transformation. B, plasmid DNA can be taken up by bacteria without being degraded. C, plasmid DNA carries a gene for resistance to specific antibiotics. D, recombinant DNA in bacteriophage vectors substitutes for a protein of the original chromosome. And E, insertion of recombinant DNA into plasmids recircularizes the plasmid. Okay, ready? Go ahead and vote. Okay, the answer is A. So I guess that we should just read through that a little bit. This is probably, in a way, minutia, but you know, if you know it, then you're ready for those exam questions. When a bacteriophage is used to introduce DNA into bacteria, the technique is called transfection. A bacteriophage is, you know, a bacterial virus, and it infects the bacteria. And if you're using it to transfer a piece of DNA, that the words are combined together, the transfer and infect, and so it's called transfect. Transform is what you do if you introduce a naked piece of DNA into a bacterium. You transform by giving it a new phenotype, and the new phenotype usually is with a plasmid, and the plasmids carry genes for antibiotic resistance. And so the new phenotype that bacteria acquire whenever you take naked DNA that contains within it a gene encoding an antibiotic resistance protein, like for ampicillin, you transform it into a new phenotype. So transformation is used to describe what happens with giving a new phenotype with naked DNA. Transfection describes infecting and therefore transforming with a new phenotype. B, plasmid DNA can be taken up by bacteria without being degraded. Well, yeah, of course it can be taken up without being degraded. Otherwise, you wouldn't generate more plasmids and you wouldn't transform the bacteria. And yes, plasmid DNA carries a gene for resistance to specific antibiotics. Recombinant DNA in bacteriophage vectors does substitute for a portion of the original chromosome, and plasmid is a circular piece of DNA, and with a molecular scissors that you use, you take that circle, you cut it, and it makes it linear, and then you insert a piece of recombinant DNA into it, and it recircularizes. And it's only the circular form of plasmids that will transform bacteria. Okay, next question. This figure shows the base pairing of a short duplex DNA molecule. Letters represent the 4 common bases present in DNA. So you see there's the double-stranded piece of DNA. The top strand is 5' to 3' left to right, and the bottom strand is right to left, so anti-parallel. The type 2 restriction endonuclease, known as BCL1, will cut symmetrically within the 6 base pair palindrome of dyad symmetry that occurs within which of the following positions of the DNA molecule? A, these are all 5' to 3'. Choice A is CCC-GGT. B, CC-GGTT. C is CGGTTG. D is TTGATC. And E is TGATCA. If you're ready, go ahead and vote. Okay, so we need to talk about that a little bit. So what this is testing is whether or not you understand the words palindrome, dyad symmetry, and whether or not you understand the concept that enzymes like BCL1, do you understand the concept that they cut DNA that is palindromic or that has dyad symmetry? And the only one of the sequences here that has this so-called palindromic or dyad symmetry is choice E, TGA-TCA. Here it is shown pictured over here on the right in this little box. The position of the cut that the enzyme makes is a staggered cut, but that's not really so important. What's important is the concept of dyad symmetry. If you read the top strand left to right, it reads TGA-TCA. If you read the bottom strand right to left, what does it read? TGA-TCA. That's what dyad symmetry means. That's what palindrome means. It reads the same forwards as it does backwards. Most type 2 restriction endonucleases, such as BCL1, recognize sequences that are palindromes. Most, and there are hundreds of these. Virtually every species of bacteria makes at least one kind of restriction endonuclease. Not all are like that, but most are, so it's an important concept, okay? So that's just an example of one of the pieces of molecular scissors that molecular biologists use to make recombinant DNA. So with things like restriction endonucleases and other enzymes known as ligases, which are the molecular scissors and tape, you can make recombinant DNAs. And one of the important things that molecular biologists do with that is to create libraries, genomic DNA libraries or cDNA libraries. So this is an example of how one might create a genomic DNA library. This represents a single strand of human genomic DNA, and that piece of genomic DNA might be cleaved into small pieces with some restriction nuclease, like BCL1 or some other nuclease, that breaks that piece of DNA up into lots and lots of fragments. The blue, the green, the yellow, the red, the purple represent specific genes, and the gray represents DNA in between those genes. But in this library, the DNA in between the genes, as well as the DNA that represents the genes, ends up as small fragments. Millions of small fragments that can then be inserted with ligases into, in this example, inserted into plasmid DNA to make a new circular recombinant DNA molecule, some of which have the genes, although it doesn't show it here, but other pieces have the spacer DNA. So all of the DNA is in the library. Take that library, introduce it into plasmids, introduce the plasmids into bacteria, grow the bacteria, clone the bacteria, and now you have a so-called library where each bacterium, each clone of bacteria, has in it a plasmid that came from a single region of the genome. In this case, the green gene, the yellow gene, the red gene, the cyan gene, the purple gene, and all of the spacer DNA in between. It's all there somewhere in this library. But there are also cDNA libraries, and there is an important difference between a cDNA library and a genomic DNA library. The genomic DNA library, represented over here on the left, as we just talked about, you take the entire DNA, you break it up into small pieces, you clone those small pieces, so every piece of DNA, whether there's a gene on it or not, is represented in the library. To make a cDNA library, what you do is to isolate RNA from some cell or tissue or organism. What has happened is that the genes encoding gene A and gene B were copied into RNAs, and the intervening sequences were spliced out to make mature RNAs. So here is the one RNA from gene A, and many more copies of the RNA from gene B, because in this example, gene A is expressed at a lower level than gene B. And so you use reverse transcriptase, an enzyme that takes RNA and uses it as a template to make a DNA, and insert that complementary DNA, or that cDNA, into bacteria and clone that. And so what you end up with now are clones that are present in the proportion as the original RNA. So gene A, 1 copy, whereas gene B, 1, 2, 3, 4, 5 copies. So there would be 5 times more copies of gene B RNA than gene A RNA. Not only that, a cDNA library then has only representatives of the messenger RNA. None of the intronic sequence, that's all gone. None of the DNA between genes is present. That's all gone. So a genomic DNA library gives you access literally to the genomic DNA. If a gene is present in 1 copy, it will be present in 1 copy. Well, if 2 genes are each present in 1 copy, like gene A and gene B, 1 copy per cell, then the relative ratio in the library is going to be 1 to 1. But in a cDNA library, everything that's expressed is represented in proportion to its abundance in the cell. Does that make sense? So those 2 kinds of libraries are used for different purposes. Any questions on that? Okay. Now there are some standard techniques for detecting DNA, RNA, and protein, and one of those standard techniques is illustrated here. It's called a Southern blot. Another common technique is called polymerase chain reaction, and we'll get to that in a minute. But I just want to make sure that you know how a Southern blot works. You take DNA that has been isolated or extracted from cells somehow, you apply it to a gel and put it in an electric field so that it separates on the basis of size. So here, examples of DNA that were extracted from some cell, placed on different lanes, run on this gel that separates on the basis of size. In this lane right here, there are marker DNAs. They're added at this end of the gel, and they're run to the right-hand side of the gel here. So this is the largest piece of DNA, and the next largest, and all the way down to the smallest piece of DNA. Then you take, after the DNA has been separated, you take this gel, it's like agarose, it is agarose. You place immediately on top of it this green thing right here that represents a piece of paper that DNA will bind to, and you allow capillary action to transfer a buffer from a solution down here into a stack of dry paper towels. And so over a period of a few hours, that buffer slowly migrates through the gel and carries the DNA from the gel to that piece of nitrocellulose paper where it binds and gets immobilized. And then you go to the next step, which is to peel that piece of nitrocellulose paper off where the DNA is now bound, and use some technique that will allow you to detect some specific gene or target or whatever you're after. And in this example, it illustrates a radioactive probe that will bind to specific bands in these different lanes. You allow some period of time for them to hybridize, then you wash off the unhybridized, and you use X-ray film or something like that to visualize the positions of the markers plus the positions of the specific bands. And so if this is a sample of DNA, let's say from a liver, and you're trying to identify a gene for, let's say, pyruvate kinase, and you have a probe for pyruvate kinase, then it will bind just to that gene and light up a single band that represents pyruvate kinase in the presence of all of the other thousands of genes that are there. So if you do this process with DNA, it's called a Southern blot, and that's because a man by the name of Ed Southern was the first person that figured out how to do that. Well, since the north, south, east, and west, and if RNA is to DNA as north is to south, then the next person that came along figured out how to do this with RNA, and so they call that a northern blot. And then pretty soon somebody came along and figured out a way to do it with proteins, and north and south were already taken, so they chose west because they were in the west part of the United States. So you just have to remember that if you're detecting DNA, it's a southern. If you're detecting RNA, it's a northern. If you're detecting protein, it's a western, period. Polymerase chain reaction, another technique for detecting small amounts of DNA or RNA. This can be used for, as you probably already know, it can be used for detecting the presence of a parasite, a bacteria, a virus in a blood sample. It can be used to find who the criminal is, that somebody that created did a crime. So the way it works is that you have a double-stranded DNA and you heat it up so that the two strands separate, they melt. And you, in order to detect a specific region of DNA, you have to know the sequence of that DNA. And by knowing the sequence of the DNA, you can design short pieces of synthetic DNA known as primers. That green thing and that red thing represent primers that can then bind complementarity to this strand or this strand. So heat the DNA to, let's say, 95 degrees. That separates the strands. Allow, okay. Cool the DNA to, let's say, 60 degrees, and that allows these primers to bind. Warm the DNA to about 72 degrees, and that allows a heat-stable DNA polymerase to extend from those primers and make a second strand of DNA, that and that. And then you repeat that cycle. So here is the first cycle. You repeat that cycle a second time. The primers again bind to the separated strands. They extend the separated strands, and then you repeat that. And you continue that. Probably a typical number of cycles is 30 or 40 cycles. Each time you repeat that, the target DNA, the DNA between those two primers, doubles. So right here, there are one copy of each of the two strands is made, the green and the red. That's after the first cycle. At the end of the second cycle, you now have one, what do we have? 1, 2, 3, 4 new copies. At the end of the third cycle, we have 1, 2, 3, 4, 5, 6, 7, 8 new copies, and so on. So 2 to the power n, where n is the number of cycles. So 2 to the 30th power, if you get out your little calculator and do that calculation, is an amazing, it's a huge number. So this is a very powerful technique for detecting small amounts of DNA or even cloning pieces of DNA. It's a typical way that you detect trinucleotide repeats and differences in different DNA samples. For example, if you're trying to detect what we call restriction fragment link polymorphisms or triplet repeats like in Fragile X syndrome, here's an example of 2 pieces of DNA, one with, who knows, maybe that's 10 repeats, and here's one with 5 repeats. And the primers flank those repeats. Once you've amplified those DNAs, you get products that are 2 different sizes, the long size for this one and the shorter size for this one, so you can diagnose. Here's another example, a forensic question. In this case, the perpetrator. Here's the criminal, a forensic sample that was taken from the victim of a crime. And here are 3 individuals, individual A and B and C, who are suspects for having committed that crime. Let's say it's a murder or a rape or something like that. We have obtained some DNA samples from each of those. So what's being done here is PCR that represents 3 different regions of DNA from each individual. Locus 1, locus 2, and locus 3, and you see that each individual has different numbers of repeats flanking or inside those primer targets. Amplify the DNA from individuals A and B and C and run them on gels. You can see from individual A, the red is locus 1, the cyan is locus 3, and the green is locus 2. See how individuals A and B and C are all different. And there's the forensic sample out there. Which one does it match? It matches individual A. So that suggests that individual A then is the perpetrator. Okay. So that ends really any lecturing that I want to do, and from here on out, let's just practice questions. And I think that you can vote on each one of those, and we will just get through as many as we can until our time runs out. There are numerous disorders that result from defects in hemoglobins. The class of hemoglobin disorders known as the thalassemias are caused by a reduction in the level of synthesis of one or more of the globin proteins, increased synthesis of adult globin genes during fetal development, mutations in the globin genes resulting in structural variants of these proteins, synthesis of abnormal fetal globin genes during embryonic development, or E, the presence of an abnormal fetal hemoglobin in the adult. Go ahead and vote. Good. 60% of you got that correct. Thalassemias are a reduction in the level of synthesis of one or more of the globin genes. And so alpha and beta globin are present in different, in something other than a 1-to-1 proportion, and that's what causes the anemia. Now I have a few here. Most of what I've talked about so far, or all of what I've talked about, is molecular biology, but there's certainly going to be some biochemistry on here. And so here's an example of a biochemistry question. The rate-limiting step in glycolysis occurs at the step catalyzed by glyceraldehyde 3-phosphate dehydrogenase, phosphofructokinase 1, phosphofructokinase 2, phosphoglycerate kinase, or pyruvate kinase. This is an example of a question that will certainly be on your test. Oops. Looks like you need to review that. The point to be made here is that metabolic pathways all have key regulated steps known as rate-limiting steps. They are typically the slowest step in the reaction, and it's important for you to review that for biochemistry. So phosphofructokinase 1 is a key regulated step of glycolysis. Next question. Under conditions of anaerobic glycolysis, the NAD required for glyceraldehyde 3-phosphate dehydrogenase is supplied by a reaction catalyzed by glycerol 3-phosphate dehydrogenase, alpha-ketoglutarate dehydrogenase, lactate dehydrogenase, malate dehydrogenase, or pyruvate dehydrogenase. Vote now. So 55% of you got it right, 45% of you need to review. Okay, more on pyruvate dehydrogenase. Pyruvate dehydrogenase activity is regulated by its state of phosphorylation. The activity of the kinase, which catalyzes the phosphorylation of pyruvate dehydrogenase, is increased by acetyl-CoA, AMP, cyclic AMP, coenzyme A, or NAD. Vote now. Oops. So, just briefly, what does pyruvate dehydrogenase do? It converts pyruvate to acetyl-CoA. Typically, enzymes like this are regulated allosterically. Many of them are inhibited by their product. So acetyl-CoA is the direct product. If acetyl-CoA builds up, it says, okay, everything upstream, quit making acetyl-CoA. I got to catch up. So it inhibits things upstream. And in this particular case, the way acetyl-CoA works is to activate the kinase, which phosphorylates pyruvate dehydrogenase and inhibits it. I put this up here just to say, okay, this is an important regulated step. You need to review it. Next question. Synthesis of glycogen is inhibited in hepatocytes in response to glucagon stimulation, primarily as a result of a decrease in the level of phosphoprotein phosphatase, decrease in the level of phosphorylated phosphorylase kinase, decrease in the level of phosphorylated phosphoprotein phosphatase inhibitor 1, increase in the level of the dephosphorylated form of glycogen synthase, or E, an increase in the level of the phosphorylated form of glycogen synthase. Vote now. So what's going on here is glucagon stimulates an increase in cyclic AMP. Cyclic AMP activates a protein kinase, right? Protein kinase A. That stimulates the breakdown of glycogen and inhibits the synthesis of glycogen. So hopefully, with that in mind, it's easy to then figure out that choice E is correct, because what's happening is increased phosphorylation. The other limb is choice B up here, the level of phosphorylated phosphorylase kinase. That's the enzyme that breaks down glycogen. But it can't be a decrease in the level because protein kinase A is activated. It's an increase in the level So you increase that and increase the phosphorylation of glycogen synthase. Looks like you need to review that, huh? In diabetes, the increased production of ketone bodies is primarily a result of a substantially increased rate of fatty acid oxidation by hepatocytes, an increase in the rate of the citric acid cycle, decreased cyclic AMP levels in adipocytes, elevated acetyl-CoA levels in skeletal muscle, or increased gluconeogenesis. Vote now. Good. I thought you'd probably get that one. Okay, let's do some more genetics here. Fragile X syndrome is characterized by accumulation of copper leading to formation of Kaiser-Fleischer rings in the eyes, hypoketotic hypoglycemia and metabolic acidosis, isovaleric acidemia, severe metabolic acidosis, and neonatal fatality, mental retardation of severity linked to the level of trinucleotide repeat expansion, and E, very long-chain fatty acid accumulation and myelin defects. Vote now. Good for you. I probably should have left that question out, but I'm glad I left it in because it gives you a little bit of confidence, right? Okay, one mechanism by which RNA tumor viruses can cause cancer is by carrying into infected cells a protein that turns off the expression of tumor suppressor genes, by restricting the function of cellular DNA polymerase, the action of reverse transcriptase induces overexpression of growth-promoting genes, to alter plasma membrane architecture upon infection, and E, to integrate near a growth control gene and alter its expression. Vote now. Oops. Okay, so an RNA tumor virus, these are viruses that are called oncornoviruses sometimes, as you know. So they have an RNA genome. They encode a reverse transcriptase, an enzyme that's capable of converting the RNA sequence into a DNA copy, and that DNA gets integrated at random sites in the host genome. That's an important aspect of the way these kinds of viruses work. So whenever you integrate anything into the DNA, that's a mutation. Would you agree with that? And so we are looking here for a feature that's mutagenic. In a way, it does carry into infected cells of proteins that turns off the expression of tumor suppressor genes. That's not very common, these other things, but the key is integrates near growth control genes, or in some cases integrates and carries in their own gene like RAS or MYC or FOS or something like that. In this particular example, this is a class known as the INT, I-N-T, for integration. Next question. Which of the following is quantitatively the major contributor to routine clinical measurements of circulating plasma cholesterol concentrations? I bet everybody gets this one. Chylomicrons, high-density lipoproteins, intermediate density lipoproteins, low-density lipoproteins, or very low-density lipoproteins. Vote now. All right, good for you. D is correct. Low density lipoproteins, that's the bad cholesterol, I guess, but it is the major cholesterol carrier. Which of the following statements best characterizes ATP synthase? It couples ATP export from the mitochondrial matrix to ATP synthesis. It is a soluble protein found inside the mitochondrial matrix. Its catalytic function is to synthesize ATP in a reaction driven by a chemiosmotic potential. Oligomycin binds to ATP synthase, directly preventing ATP export. And the low proton concentration outside the inner mitochondrial membrane establishes an electrochemical gradient that drives ATP synthesis. I vote now. Well, you're on the right track with choice E, but see how choices E and C are similar? In one case, proton concentration is low outside the inner mitochondrial membrane. In the other case, it's high. What happens? The electron transport chain pumps protons out of the inner mitochondrial matrix into the space outside the inner mitochondrial membrane. Protons are high there, and then they flow back in and turn this motor that makes ATP. So you chose E. You're thinking kind of right, except it's not a low proton concentration outside, it's a high proton concentration outside. So C, its catalytic function is to synthesize ATP in a reaction driven by a chemiosmotic potential. Do you understand? Truth be known, when I first saw that, I answered the same thing, and I go, of course, duh. The net conversion of carbons from fat into carbons of glucose cannot occur in humans because fat oxidation occurs in the mitochondria and gluconeogenesis occurs in the cytosol. States of catabolism and anabolism are never concurrently active. Storage of fats occurs in adipose tissue and gluconeogenesis occurs in liver and kidney. The carbons of acetyl-CoA from fat oxidation are lost as carbon dioxide in the TCA cycle. The carbons of acetyl-CoA from fat oxidation inhibit conversion of pyruvate to oxaloacetate. Vote now. Well, the answer is D. 36% of you answered D. You know, in a way, I hate this question, but it always shows up, it seems like. It's almost a trick question, but that's the answer right there. So both carbons of acetyl-CoA that enter the TCA cycle eventually get converted to CO2, so there can never be any net production of fat from carbohydrate because of that. Yeehaw. Which of the following is associated with type 1 diabetes? Decreased insulin receptor response to insulin binding, impaired glucagon-dependent inhibition of glycolysis leading to hyperglycemia, decreased glucagon secretion leading to hypolipidemia, elevated insulin secretion leading to severe hypoglycemia, or elevated glucagon secretion leading to hyperlipidemia. Vote now. Oops. Well, I would have guessed that most of you would have gotten that one. So type 1 diabetes is the form of diabetes that's insulin dependent, right? And under those circumstances, glucagon levels are elevated. Okay. The signal transduction protein RAS has been shown to play a role in the genesis of certain types of cancer. This effect is due to alterations in the sequence of this important regulatory protein. RAS belongs to the family of signal transduction molecules that are A, activated by GTPase activating proteins, or GAPs. B, guanine nucleotide binding proteins, termed G proteins, nuclear transcription factors, protein serine threonine kinases, or receptor tyrosine kinases. Vote now. This is up here for an important reason. Yesterday, Dr. Graham talked to you a little bit about the pathology associated with different kinds of oncogenes. There are a variety of different classes of those different oncogenes, and they're all listed here. Some of them are mutated forms of transcription factors. Some are mutated forms of serine threonine kinases. Some receptor tyrosine kinases, some the GAPs, and some guanine nucleotide binding proteins. And I think it's important for you to get in your head a general idea of what different oncogenes belong to which class. Like MYC is a transcription factor. RAS is a G protein. It switches on and off with G. The reason choice A is not correct is because it says activated by a GTPase activating protein. Well, it's actually inactivated. RAS is active when it's bound to GTP, and GAPs come along and activate the enzyme activity that then breaks down GTP to GDP, and that immediately turns the RAS switch off. That's common to all of these G proteins. RAS is just one of many examples of G proteins. Anyway, I think you'll do yourself a big favor if you study and learn the different classifications then of the oncogenes and commit them to memory, or at least to understanding. How are we doing for time? Oh, we're okay. Some more genetics. Mosaicism refers to a condition in which an individual has two or more genetically distinct cell types. It occurs with a frequency of approximately 30% in females who harbor gonadal dysgenesis, sexual immaturity, and infertility. These females are also short in stature, have webbed necks, swelling of the hands and feet, as well as cardiovascular and renal abnormalities. These symptoms are associated with achondroplasia, Duchenne's muscular dystrophy, Fragile X syndrome, Klinefelter's syndrome, or Turner syndrome. Vote now. Good for you. That actually is probably less genetics and more pathology, huh, Dr. Graham? Some more of the same. Numerous cancers are caused by a genetic phenomenon termed loss of heterozygosity. This phenomenon led to the identification of genes termed tumor suppressors because it is loss of their function that leads to cancer. Which of the following has been shown to result from defects in a tumor suppressor gene? Creutzfeldt-Jakob disease, Crouzon syndrome, Huntington disease, Li-Fraumeni syndrome, or Prader-Willi syndrome? Vote now. So this question really requires that you know two general kinds of information. You know some genetics, you know what loss of heterozygosity means, but it's probably more testing your knowledge of pathology, right? And so Li-Fraumeni syndrome is a syndrome in which a tumor suppressor gene, Dr. Graham mentioned this yesterday. Do you remember which tumor suppressor gene is mutated? This is probably the most important tumor suppressor gene. It's P53. P53 mutations occur in probably more than half of certain types of human cancers. So P53 is important to know about. So this particular syndrome is just one of a whole host of different kinds of cancers or conditions in which there are P53 mutations. Loss of heterozygosity. Let's briefly just discuss that, and I'm sorry I don't have any slides to help illustrate it. The idea is this, is that all tumor suppressor genes are recessive mutations. As far as I know, all of them are recessive mutations, because what is required is a normal function of the gene. So P53 is a protein that's often called the guardian of the genome. It monitors whether or not there's DNA damage. If there's DNA damage, then P53 activates a whole host of processes that cause a cell to either repair the damage or kill itself. So fix the problem or die. P53 is the guardian of the genome. So if one of the two copies of P53 has a mutation so that it is non-functional, the normal copy is still functional, and so the genome is protected. That would be a heterozygous individual. So you might see individuals that have one normal and one abnormal copy of a P53 gene, and they don't have a tumor. They're heterozygous. But suddenly an individual comes along and develops a tumor, and you take a biopsy of the tumor and you analyze the P53 gene, and you find that both copies of the P53 gene are mutated. So now you have a homozygous condition, homozygous for a mutation in the P53 gene. Whereas before the tumor formed, that individual was heterozygous for P53. Now they're not heterozygous anymore. They have lost heterozygosity. Does that make sense? That's very, very typical with before tumor and after tumor in individuals with mutations in tumor suppressor genes like P53, loss of heterozygosity. That's an important concept. A polymerase chain reaction can increase the sensitivity of certain genetic tests. Necessary components of a polymerase chain reaction include all of the following except the DNA to be amplified is denatured in the presence of an equimolar ratio of primers. A heat-resistant DNA polymerase is used for strand synthesis. Multiple heating and cooling cycles are required for amplification of the DNA. And the sequence of the segment of DNA to which primers will bind must be known. Vote now. Well, this is something I didn't emphasize earlier, but the primers have to be added in thousands of fold molar excess of the target, millions of fold molar excess, not equimolar amounts, because there have to be enough primers for every cycle. Every piece of DNA in each cycle needs to be bound by a primer that then gets extended, okay? We're getting a little short on time, and I would like to let me move forward quickly and find a genetic question. Okay, this is the last question, I think. Okay. Cystic fibrosis is an autosomal recessive disease with an incidence of 1 per 1,600 in the Caucasian population. What is the frequency of the cystic fibrosis gene? 1/4, 1/20th, 1/40th, 1 in 200, 1 in 400. Vote now. Good, most of you got that right. 1 in 40. I said this was the last one, but the next one is linked to this to help you understand the answer. And what I want you to do is in your studying, learn, if you don't already know, learn what the Hardy-Weinberg law is. Now this is a little more tricky of a question right here. Actually, I'm going to go to the next one. Tay-Sachs disease occurs almost exclusively among Ashkenazi Jews with an incidence of 1 in 3,600. The frequency of carriers of the Tay-Sachs gene, which can be calculated by using the Hardy-Weinberg law, and there it is mathematically expressed right there, p squared plus 2pq plus q squared equal 1, is which of the following? 1/4, 1/30th, 1/60th, 1/600th, or none of the above? Now before we vote, hang on, don't vote yet. Let me give you just a few more seconds. Too late. Please vote. So B is the correct answer. So let me walk you through that. Understanding the Hardy-Weinberg law is the key to understanding the previous question about cystic fibrosis or most of these other things. What p squared, 2pq, and q squared are all about, p is the frequency of the normal gene, q is the frequency of the abnormal gene. So the number of homozygote normal, homozygote affected, and heterozygous carrier have to add up to 1. You know, the normals, the carriers, and the afflicteds have to be 100% of the population. And this term describes it right here. Well, they say the incidence is 1 in 3,600. So in other words, q squared is 3,600. So what is q? It's the square root of 3,600. So the frequency then of that occurring is the frequency of the gene is 1 in 60. But the question is what's the frequency of carriers? The frequency of carriers is right here. It's 2 times p times q. Well, p is almost 1. So for all practical purposes, that's 2 times 1, which is still 2, times q, which is 1/60th. So it's 2 times 1/60th, which is 1/30th. Do you follow? Okay. With that, I need to shut it off, but learn that law. That's important. Thank you. 5 minutes.
Yeah, thank you. We will take a brief 5-minute break, so please return promptly in 5 minutes as we'll need to get started. Thank you.